In this lesson, we'll do a quick run-through of how the brain creates and uses a memory, from encoding to storage to consolidation and retrieval, and the role of the hippocampus. Here we see the human brain and a cutaway showing the hippocampus. We have one on each side of the brain. Of course, brains are made of neurons, and they show this property of plasticity. Plasticity is the ability of neurons to change the strengths of their connections, what we call synapses. And changing synapse strength, that's the biological basis of memory formation. We can use a simple icon to represent a synapse here, and to show that a synapse has grown stronger, we can just sort of make the terminal bigger. And if we're detailed, maybe show more neurotransmitters there. And so a general principle of brain function, neuron function, is that neurons, cells that fire together, wire together. So if these two neurons were involved in a learning episode, the conditions are set up so that the synapse can strengthen. Now, since neurons show plasticity, memories can be weak or strong. Now here we see a series of pictures, and we see synapse is getting stronger as you go down the diagram here. Now, if this were a school setting, this might be when we're studying multiple times. As we study repeatedly, we give the memory trace neurons a chance to build stronger and stronger synapses. As we do so, the memory trace is going to get stronger as well. So a strong memory trace is strong because it is composed of neurons that have strong synapses amongst them. A weak memory trace would have neurons with only weak synapses. Now, scientists think our long-term memories are mostly stored in the cerebral cortex. It's a large structure with lots of neurons, lots of synapses to store information. So we can liken it to the hard drive of a computer. Now, there are other aspects of memory stored in subcortical structures like the amygdala and other places for emotional aspects of memories and so on. But for most of what we're thinking about, episodic and semantic memories, it's going to be the cerebral cortex that will be the long-term storage system, the hard drive of the brain. Now, computers store information differently from brains. Here's the hard drive of the computer. Here's the RAM. We'll associate that with the working memory system. And when we click Save, we're working on a document and we click Save, the computer is writing the data to the hard drive. But importantly, all that information is stored with the same strength. And incidentally, when we open up a document, then the computer is going to read the data from the hard drive and then display it on our monitor. But the important point here is that unlike brains, computers store information with the same strength. That's not true for brains. We'll be learning that the hippocampus is, in a sense, as it performs its function, it's writing the data to the hard drive, to the long-term memory system, and we call that process consolidation. Well, the amount of consolidation determines the strength of the memory. So in human brains, we can have weakly stored memories and strongly stored memories, unlike a computer. Now, there's another memory system we want to take note of, and that's the working memory system. On our map of the brain, there are a couple of terms here, thought, forethought. And so we're going to associate those frontal cortex systems with what we're calling the working memory system, or we're just going to put a box here. And whatever information gets into the working memory system in this box, that'll be what we're sort of consciously aware of. Now, we've learned that in the back part of the brain, we've got three major sensory systems: vision, touch, hearing. We've got language comprehension systems here. So whatever processing is happening in the back can be shared with the working memory system, and we can become aware of what we're seeing and what we're feeling on our skin and what we're hearing and language in our environment, etc. So in the rest of the lesson, we'll see some kind of box in the front part of the brain representing the working memory system, and whatever is being shared with that system is what we're currently aware of, what we're conscious of, what we're currently thinking about or processing or perceiving. So now it's time then to go through these processes: encoding, storage, consolidation, and retrieval. And we'll use the example of witnessing a solar eclipse, and we saw that example in a previous lesson. In the remainder of the lesson, we'll need to be familiar with a couple of icons. Here we see the surface of the left side of the cortex here, and we've got two icons to take note. The red patch here, that's going to be an icon for a population of neurons being activated in a region of the brain. In this case, the visual cortex is being activated, but not the touch or the hearing cortex. Now, some of those neurons will go on to take part in forming a memory trace. And for a memory trace, we're going to have a different icon, and that's the paper with the pen applying ink. So that'll be an icon for a memory trace forming in a certain location in the brain, and the ink represents synapses getting stronger between neurons involved in the memory trace. Okay, let's begin then. The first stage in creating a memory is the encoding stage. And here we have regions of cortex that are actively responding to the specific characteristics of the event. So the visual cortex is processing what the eclipse looks like. The touch cortex processing what the sand feels like and the wind on the skin. And the auditory cortex processing the sound of the crowd and the sound of people saying things during the event. So we've got the cortex is responding with a certain pattern of neural activity that is specific to the event. In previous lessons, we've used this video to give us an intuitive sense of what's going on in a region of cortex that is being activated by some input. So here we see these flashing lights here are neurons that are generating electrical signals, but they've been genetically engineered to emit light. So it gives us a way of directly seeing sort of the activity of neurons, of a population of neurons in a region of cortex. So that's what we want to think is happening in these different areas of the brain that are processing the event. A population of neurons is being activated. Now, the axons from those neurons can carry electrical signals all the way up to the frontal cortex. So each of these areas of the cortex can communicate with the frontal cortex, and that allows what we're seeing and feeling and hearing to reach the working memory system. And when it does so, we can become conscious of the event. We're aware of what the eclipse looks like and feels like and sounds like. So sharing information with working memory is how an experience can be conscious. Now, but to make a long-term memory of the event, information must also be shared with the hippocampus. So these cortical regions that are involved in sharing with the working memory system are also sharing the results of their processing with the hippocampus. So we have to think of axons branching to carry information to two different brain locations. So the cortical regions are also sending information that converges in the hippocampus. Let's look at that again. So here we have what it takes to become conscious of the event, but we've got to involve the hippocampus to make a long-term memory of the event. We saw in a previous lesson a diagram that looks like this, where we break down what the encoding process is and storage process. So responding to an event is not enough to make a memory. Neurons must change their shape in a way that can represent the learned information. So over here we see some cortical neurons, and some of them, the orange ones, are being activated by the event. This is the encoding stage. Notice the blue neurons are not active. But after that, after the event is over, we're going to have a period of biological growth. And that's the storage stage. So now we're getting into the second stage. What's going to happen here is some of those neurons that were activated by the learning event are going to grow stronger synapses. This is happening after the event. Notice the orange neurons are not currently active. And the growth of these synapses, that's what we're identifying as the memory trace for the event. So this is what has to happen if we're going to make a memory of the event. It's not enough to just have neurons respond. They have to change in some way that sort of records the specific characteristics of the event. And we also saw in a documentary this time-lapse footage of the process of memory trace formation. So when we play this, we see the red dendrites of the target neuron, and we see the green axon terminals, and the bulges would be synapses. We see the growth of new synapses. This is the storage of a memory trace. Here are two neurons that are strengthening their synaptic connections, and that change, that biological change, is the memory trace that represents some learned information. In this case, the animal was learning to associate a touch with a shock. This was a sea slug, and these are two neurons, and they did time-lapse photography. After the training, they saw this change in synapse strength. There were more synapses being created. This is the formation of a memory trace. Now we're going to use an analogy. We've already seen that the icon for a memory trace is a piece of paper with applied ink. And the idea here is that when we're making memory traces, we are sort of printing memories into the circuitry of our brains in the cortex and in the hippocampus. So a weak memory would be a piece of paper with very little ink, and then with more ink, that's a stronger memory trace, and even more ink, a really strong memory trace. So as a result of this growth process, synapses are growing stronger. We've got memory traces being formed in the cortex as well as the hippocampus. And again, the memory traces in the cortex are representing what the event looked like, what the event felt like, what the event sounded like. But the hippocampus is linking all of them together to kind of form a unified sort of memory trace that kind of represents the pattern of cortical activity unique to that particular learning event. So the sensory qualities of the learning event are linked together in the hippocampus, and this hippocampal memory trace represents the pattern of activity in the cortex. Again, just to review our vocab, a memory trace is the biological change that occurred in the brain that represents the acquired information. The biological change involved the growth of stronger synapses among neurons in these different locations of the brain. So the full memory trace for the event includes not only the cortical memory traces, but also the hippocampal memory trace that links them. Again, plasticity is what allows this to happen. Neurons can change synapse strength, and when they do so, we call that the formation of a memory trace. So far then, we've covered the encoding stage, where a population of neurons is being activated by the learning event. And then after the event, there's biological growth of synapses. That's the storage phase, and the biological changes that happen there, that's the memory trace for the event. And so now we're ready to sort of more broadly give a definition of what is a memory. A memory is a pattern of strengthened synapses among neurons activated by the learning episode and represents the learned information. So now let's continue with the story. After one learning episode and after the growth of the relevant synapses, we've got memory traces stored in the cortex and the hippocampus. But notice we're indicating that the cortical memory traces are weak. There's not a lot of ink applied to the paper there, whereas the hippocampal memory trace is strong, lots of ink. Now, why would that be the case? Well, scientists would explain it this way, that the hippocampal neurons show higher plasticity than cortical neurons. They can make large increases in synapse strength after one episode. So here we have the strength of the synapse before learning and after learning, a major increase in synapse strength. This makes the hippocampus a structure that is a fast learner. It's got a population of neurons. They're like the elite athletes of the brain. They are very responsive. One learning episode and they can increase synapse strength dramatically. By contrast, in the cortex, those neurons show lower plasticity, so they make smaller increases in synapse strength after one learning episode. So notice we go from a weak synapse to a slightly stronger synapse after learning. We would say the cortex is a slow learner. So this is what would explain why we have different strengths of memory traces in the cortex and in the hippocampus. But while it's reasonable to explain the weakness of the cortical memory traces and the strength of the hippocampal memory trace by referring to differences in plasticity, what really is the evidence that the cortex is only making weak memory traces to begin with? And that evidence comes from people like H.M. and Clive. Now, Henry Mollison lived in the 1950s, and he had surgery to treat epilepsy, and the surgeons removed his hippocampus on both sides. Clive had a viral infection, and his hippocampus was damaged on both sides of the brain. And the study of people with hippocampal damage shows that without a hippocampal memory trace, the brain evidently has difficulty finding and reactivating cortical memory traces. And so it's from this kind of observation that scientists suggest that the initial cortical memory traces are too weak to find and reactivate. So H.M. and Clive, they have an intact cerebral cortex here, so they can see things and feel things on their skin and hear things, and they can be conscious of these experiences in their life. But evidently, the memory traces that are made in the cortex are insufficiently strong. So they're too weak for the cortical retrieval systems to find those memory traces and reactivate them. And so these individuals appear to us as being incapable of making new episodic and semantic memories. So the idea then is that the initial cortical memory traces are weak, and we won't be able to retrieve them with the cortical retrieval systems, and that's why we're going to need the hippocampal memory trace down here to help the brain find and reactivate those cortical memory traces that are weak initially right after learning. So the hippocampal memory trace will be used to selectively activate the appropriate cortical memory traces. All right, it's time to move on to the retrieval phase of memory formation and use. Now, in order to retrieve a memory, the memory trace neurons must be reactivated. So the brain is going to attempt to reactivate these cortical memory traces and then load them up into working memory, and then we'll enjoy a retrieved memory. Now remember, a retrieved memory often starts with some kind of cue. Somebody asks us, hey, you remember what happened yesterday, the eclipse? And then the brain is going to have to attempt to find the relevant memory traces and reactivate them. So now let's tell the story of what's going on in the brain when we retrieve a memory. So let's say the eclipse happened 3 hours ago, and somebody says, do you remember the solar eclipse on the beach? Well, that information is being processed by my auditory cortex and the language areas of the brain, and I'm conscious of the word solar eclipse. But the auditory cortex is going to be activating the hippocampus as well. And now we're going to have a chance to reactivate the hippocampal memory trace, which included aspects of what the solar eclipse event sounded like, including the word solar eclipse. So now the cue, right, the person asking us to remember something, the cue is going to activate that strong hippocampal memory trace there. And now, because the hippocampus and cortex have two-way communication, the hippocampal memory trace can reactivate the appropriate cortical memory traces. And then once the cortical areas are activated, they will share their information processing with the working memory system, and we will enjoy a retrieved memory. We'll be conscious of the event, what it looked like, what it felt like, what it sounded like. Now, it might not be as vivid as seeing it right in front of our eyes, but we will have the experience of a relived moment in time. So when we're retrieving information, retrieving an episodic memory in this case, our brain is attempting to recreate the pattern of neuron activity that occurred during the original learning event. If we're successful, we will have a retrieved memory. Notice that during retrieval, we have a population of cortical neurons that are being reactivated. We can use that video to remind us of sort of what it might look like for neurons to be reactivated. Well, it's the same set of neurons that were activated during encoding. And this, after all, is why we use the word reactivation during retrieval, because we're turning on that same set of neurons that was activated during the encoding stage of the process. Let's remind ourselves what's happening at the level of neurons. During encoding, we have a population of neurons that are being activated by the learning event. During the storage stage, a subset of those neurons are going to grow stronger synapses with each other, forming the memory trace. And that sets up this population of neurons to become the memory trace neurons. And then during retrieval, we're going to attempt to reactivate those very same memory trace neurons. And if we're successful, then we can retrieve the information. So when we retrieve a memory, we're attempting to recreate the pattern of neuron activity that was present during the encoding stage, during the learning event. So here we've told the story then. We've encoded information of the event. We grew some stronger synapses to create a population of neurons that forms a memory trace of what it looked like, what it felt like, what it sounded like. And then we've retrieved that memory by reactivating those memory trace neurons. This, by the way, was the solar eclipse in La Serena, Chile in 2019, and more recently in Texas in 2024. This using a different camera, a different lens. Get a picture like this. It's a beautiful natural spectacle. So for recently learned information in which we only have weak cortical memory traces, our brain needs the help, the assistance of those strong hippocampal memory traces to find and reactivate those cortical memory traces. And this is what we have concluded from the research on these individuals with damage to the hippocampus. Without a hippocampus, the cortical retrieval systems just can't find and reactivate those weak memory traces. Now, interestingly, the other pattern of memory performance in these individuals is that remote memories were retrievable in these patients. So here the idea is that the cortical memory traces somehow got strengthened to the point where the cortical retrieval systems could access them and retrieve that information. So for well-stored memories in the cortex, we don't need the assistance of the hippocampus to retrieve them. And of course, that's the key to making long-term memories, is to make strong cortical memory traces, whether we're talking about episodic or semantic memories. So the question that remains then is, how are cortical memory traces strengthened? And we will see there is a starring role for the hippocampus, again, in that part of the story coming up next.